Bismillahirrahmanirrahim. Assalamu alaikum to all our viewers. This is our educational channel Learning Biology Online, and we are here with the continuation of our previous lecture that were on cell, where we will continue to discuss the different organelles and how they are performing their functions within a cellular environment. So by the end of this lecture, we hope you will be having a very good understanding about what lysosomes, peroxisomes, vacuole and glycoxysomes are and how they function within a plant or an animal cells, and what is their key role in the different sorts of environment that in which they are working in. Starting off with lysosomes, so these were first discovered in somewhere in 1949 by a scientist named Deuve, who discovered them as single cell membrane bounded organelles, and they were rich with acid phosphatase enzyme that has the ability to break down certain macromolecules to release energy. So the process of this enzyme formation that occurs within the ribosomes, and from the once these enzymes are formed, they are packed in the Golgi vesicles, and this is how primary lysosomes come into existence within a cell. So you can see over here in the diagram this how a food particle is engulfed into the cell, and then you can see budding of these lysosomes from the Golgi apparatus, and this is how a lysosome is formed. You can see budding off from these certain vesicles from the Golgi apparatus, and these basically consist of that particular digestive enzyme inside, and you can see how they function. These lysosomes, when any food particle is going to enter into the cell, it is engulfed by the lysosome somewhere in this region over here, and then they are hydrolyzed within the cellular environment, and then the waste is then moved out of the cell through exocytosis. And this is basically that portion of the lysosomal activity where digestive vacuole or enzymatic activity occurs within the cell. This whole structure is known as the digestive vacuole because it consists of that particular digestive enzyme and the food particle, where food particle is broken down to produce energy. So apart from engulfing foreign objects, you can also see some other functions that are performed by the lysosome that involve breakdown of certain food particles to produce energy when the body is under starvation. तो जब भी किसी वजह से अगर फूड इनटेक इंडिविजुअल नहीं कर पा रहे होते तो हमारे लाइसोसोम्स क्या करते हैं कि जो स्टोर्ड एनर्जी है उनके मॉलिक्यूल्स का ब्रेक डाउन करवा देते हैं, सो दैट द बॉडी डज नॉट suffers an energy breakdown. And then you can see how lysosomes are involved in autophagy, that is a cell recycling process in which the old cells that are they undergo apoptosis and there is formation of new cells simultaneously. Lysosomes, they are also clinically significant when it comes to certain congenital diseases. So congenital diseases basically क्या होती हैं कि there is some sort of mutation within the genes, जिसकी वजह से एक specific gene क्या करता है कि किसी भी एक cell के specific component का जो या organelle का function है उसको compromise कर देता है. जैसे कि अगर हम specifically lysosomal एंजाइम की एक्टिविटी की बात करें तो दे आर वेरी इम्पॉर्टेंट इन ब्रेक डाउन ऑफ एनर्जी मॉलिक्यूल्स. तो अगर किसी भी वजह से इनकी म्यूटेशन हो गई है तो देयर कैन बी सर्टन डिजीज़ जिसकी वजह से सर्टन मैक्रो मॉलिक्यूल्स का जो ब्रेक डाउन है वो प्रॉपरली उसकी अकरेंस नहीं होगी और सेल जो है वो प्रॉपरली एनर्जी प्रोड्यूस नहीं कर सकेगा. सो यहाँ पे आप एग्जांपल्स देख सकते हैं ग्लाइकोजेनोसिस की, type 2 glycogenosis, in which there is absence of an enzyme that breaks down the glycogen that is stored form of glucose to produce glucose. So there is absence जिसकी वजह से there is occurrence of a disease, a congenital disease that is known as glycogenosis type 2 disease. Then also we have Tay-Sachs disease, in which there is absence of an enzyme that breaks down certain lipid macromolecules. So there will be accumulation of lipid within the brain, and this will cause abnormality or dysfunction in the activity of brain that has a connection with other body organs as well. Then we come to another important cell organelle, that are peroxisomes, that were first discovered within the liver and some other cell tissues in 1965 by a scientist named Deuve, that appeared to be a single membrane bounded organelle within the cytoplasm. So liver में हमारे पास different oxidative enzyme होते हैं, जैसे कि हाइड्रोजन peroxidase है, उसके अलावा glycolic acid oxidase है. So these are certain enzymes that are involved in detoxification of certain waste materials. So the peroxisomes, they are microscopic organelles with around about 0.5 nanometer of diameter, and you can see the different species in which these peroxisomes can be found, including higher plants, yeast and protozoa. So the formation and decomposition of hydrogen peroxide basically occurs within this cell. So a very significant function of this peroxisomes within the plants is the metabolic activity that involves breakdown and formation of certain objects within the plant cell. So this is the diagram over here, in which you can see vesicular like appearance of the peroxisomes right over here within this cell. So you can see they are slightly larger in size as compared to the lysosomes, and this is how they are located within a cell. Then we come to glycoxysomes, that are found at germination stage in plants. That is basically a stage in which the plant basically grows from a सीड. तो सीड का जो सेल होता है ड्यूरिंग डेवलपिंग स्टेज, उसमें उस वक्त ग्लाइकोक्सिसोम्स पाए जाते हैं, एंड दिस स्टेज द सीड बेसिकली रिलाइज ऑन stored fatty acid to provide them energy for this formation of new plants. So this is the stage at which glycoxysomes, they are present within the plant cell, and they are very important during the glycoxylate cycle, that is conversion of fatty acids into carbohydrates. This conversion is significant because it provides energy to the seeds of the plant during development. This is just a small outline of a growing plant cell, where you can see a large sized vacuole and a relatively small sized nucleus, and you can see how glycosomes are located within this structure over here, pretty much similar to the peroxisomes, but these glycoxysomes are only present during the development of a seed. So lastly we come to the vacuoles, that cover a very large portion of the plant cell and with a lot of abundance. They are single membrane bounded organelles, and these vacuoles basically develop by coalescence of certain water filled vesicles that after a mature cell of plant they appear to be a single molecule. So इनका main function क्या है कि they are basically storage components of water within the plant cell. अब चूँकि plants को different times के ऊपर water की जो है वो continuously ज़रूरत पड़ती रहती है during transpiration and during even photosynthesis. So this component of their cell is very significant during providing or formation of energy and also in their growth. Grammatically, this is how you can see a mature plant cell, where you can see a large vacuole that is present within the cell, taking almost central position, whereas the nucleus component is pushed to one side, whereas in comparison of this to the animal cell, the nucleus is largely covering the central portion, and the vacuole component is not that prominent as in case of the plant cell. So this is the end of our video. Do like our video and share it with your colleagues if you think these topics can be informative and helpful for them in understanding all the portion of the cell organelles that we have covered in this lecture. In the comment section, do mention if you have any question relevant to the topics that we have covered, and do not forget to subscribe to our channel. Click the bell button so you can receive notification of our updated lectures on regular basis. Thank you very much.